Hello friends, welcome back to my YouTube channel Nesting Padasalai. So this video is a continuation of part 2 video. So here we are going to study few important topics that are highly important for GATE examination. Okay, so today we are going to study principal type of RNA produced in cells. So these are the list of type of RNA. First RNA is mRNA, which is also called as messenger RNA, which is involved in coding of protein. Next is rRNA, which is also called as ribosomal RNA, which is forming a basic structure of ribosome and it also catalyzes the protein synthesis. Next is tRNA, which is transfer RNA. These tRNA are central element in protein synthesis. There they will be acting as an adapter between mRNA and the amino acid. Next is snRNA, which means small nuclear RNA. The main function of this snRNA is involved in splicing of your pre-mRNA. Next is snORNA, which is small nucleolar RNA, which is involved in processing and chemically modifying your ribosomal RNAs. Next is saRNA, which is small cagal RNA. These RNA are used to modify small nucleolar RNA and small nuclear RNA. Next is miRNA. These are microRNA which are involved in regulating your gene expression by blocking the translation of selective messenger RNA. Okay. Next is small interfering RNA. Okay. So the main function of this siRNA, they turn off the gene expression by directing the degradation of selective messenger RNA. Okay. So the other function of this siRNA, they are involved in establishment of compact chromatin structure. Okay. The other non-coding RNA or ncRNA are involved in functioning diverse cell process, like they are involved in telomere synthesis, X chromosome inactivation, and also few ncRNA are involved in transporting of protein into ER. Next we'll be studying some human protein that have been synthesized from the gene that are cloned in bacteria or eukaryotic cell. So this involved, here we are going to study gene farming products or molecular farming. First protein is alpha antitrypsin, which is used for the treatment of emphysema. Next is deoxyribonuclease, which is an enzyme which is used for the treatment of cystic fibrosis. Next is epidermal growth factor, which is used in the treatment of ulcer. Next is erythropoietin EPO, which is used in the treatment of anemia. Next is factor 8, which is used in the treatment of hemophilia. Next is factor 9, which is used in the treatment of Christmas disease. Next is fibroblast growth factor, which is again used in the treatment of ulcer. Next is your FSH, that is follicle stimulating hormone, which is again used in the treatment of infertility. Next is granulocyte colony stimulator factor, GCSF, which is used in the treatment of cancer. Next is insulin is used in the treatment of diabetes, but insulin-like growth factor 1 is used in the treatment of growth disorders. Interferon alpha, which is a human protein that have been cloned in either bacteria or eukaryotic cell, which is used for the treatment of leukemia. And interferon beta is used for AIDS. Interferon gamma is used for rheumatoid arthritis. And all the interferon and interleukins are used for the treatment of cancer. Interleukins are again, they are used for cancer. In addition to cancer, they are used for the treatment of immune disorder. Next is lung surfactant protein, which is used in treatment of respiratory distress. Relaxin is again a protein which is used in the treatment of, which is helping in aiding, which is used to aid in childbirth. Serum albumin is used as a plasma supplement, and both the somatostatin and somatotropin are used in treatment of growth disorder. And next is superoxide dimutase, which is a human protein which is used in treatment of free radical damage in case of kidney transplantation. Next is tissue plasminogen activator, which is used for heart attack. Next is TNF, tumor necrosis factor, is used in treatment of cancer. Next, we are going to see some few important things you should know about human genome. The DNA length of human genome is 3.2 into 10 to the power 9 nucleotide base pair. The total number of gene present is 25,000 and total number of pseudogene present is 20,000. Okay, so these things you should know. Next, we are going to see major nutritional type of microorganism. So, you should know microorganisms are classified into different types based on the nutrition. Okay, so based on nutritional type, the microorganisms are classified into photolithoautotroph, photoorganoheterotroph, hemolithoautotroph, hemolithoheterotroph, hemoorganoheterotroph. And the carbon source, energy source, electron source, and the representative microorganism all get varied. So first, if you take energy source, for photolithoautotroph and photoorganoheterotroph, since the name starting with photo, the energy source is light. And in case of hemolithoautotroph and hemolithoheterotroph, the energy source is from inorganic chemicals. Whereas in organoheterotroph, since the name contains organo, definitely the energy source is coming from organic chemical. And with regarding to the carbon source for photolithoautotroph and hemolithoautotroph is carbon dioxide since they are autotroph. For rest of all the three like photoorganoheterotroph, hemolithoheterotroph, chemoorganoheterotroph, the carbon source is coming from organic carbon. Okay. Next is regarding to electron donor. Only for this photoorganoheterotroph which contains the word organo and hemoorganoheterotroph which again has the organo, definitely the electron source is from organic electron donor. Rest of all the three organisms, the electron source is coming from inorganic electron donor. Like if you take photolithoautotroph, the representative microorganisms include purple sulfur bacteria, green sulfur bacteria, cyanobacteria, diatoms and eukaryotic algae. In this, if you take purple sulfur bacteria and green sulfur bacteria, they cannot able to oxidize water. So in case they will be selecting the electron source for them is either hydrogen or hydrogen sulfate. Okay. Next is with regarding to the representative microorganism for photoorganoheterotroph include purple non-sulfur bacteria, green non-sulfur bacteria. So the electron source for them is organic electron donor. Next, the representative microorganism for hemolithoautotroph, it is sulfur oxidizing bacteria, hydrogen oxidizing bacteria, all your methanogen and nitrifying bacteria and iron oxidizing bacteria. All of them belonging to hemolithoautotroph. Why means the electron source is inorganic electron donor because all these organisms will be oxidizing the inorganic compound like iron, sulfate, nitrate in order to derive both your energy as well as electron. So the electron source for hemolithoautotroph is inorganic electron donor and energy source is also from inorganic. Next is hemolithoheterotroph. The representative organism includes sulfur oxidizing bacteria and last one is hemoorganoheterotroph and most of the non-photosynthetic microorganism belonging to this particular hemoorganoheterotroph and generally most of the pathogenic microorganism, pathogenic fungi and many of the protists and archaea will be belonging to this particular hemoorganoheterotroph. So in case if there is any question asking archaea, the carbon source for archaea is organic carbon, the energy source is organic chemical and electron source is from organic electron donor. Okay. So thank you friends, we'll be continuing this revision series in part 4. Thank you friends, thanks for watching this video.